Hi everyone, welcome back to the Tox Lab. I'm Rebecca.
I'm Rob. So I don't know about you, Rebecca. I love watching crime dramas.
Oh, I love it.
It's a guilty pleasure of mine, for sure. But what I like more than watching crime dramas is pointing out all the bits that are just wrong.
Yeah, that is always fun when you're like in the know a little bit.
But the bit that I really enjoy is where they find a body, they look at a body, and they can instantly tell when that person has died. And we thought this week that we'd have a little look at that phenomena and how it affects us as toxicologists, and also whether there are ways of actually accurately determining when somebody has died. So, a little bit of a health warning. We are going to be talking about quite a lot of death in this episode. And also, I'm not a forensic pathologist. If there's anybody listening who is a forensic pathologist and wants to correct me on a few things I say, please do get in touch. We'd love to hear from you. So, post-mortem interval. What are we talking about? Well, we're talking about the time elapsed since somebody has died. And this can be important for a lot of reasons. It can be important in legal cases, as in the crime dramas, where somebody's found a body and they need to know when that person might have died for investigative reasons. But there's also implications for us as toxicologists. And there certainly have been cases in the past where toxicology was perhaps misinterpreted because post-mortem interval wasn't considered in the interpretation. So it can be very, very important. So how do you estimate post-mortem interval? So one of the most obvious ways is actually environmental clues. If somebody's died at home, for example, seeing when the milk in their fridge went out of date, or things like that might give you a clue as to when they were last alive. But there are a few fairly rough and ready ways of estimating post-mortem interval, aren't there, Rebecca?
Yeah.
You can look at alga mortis, or the cooling of a body. Essentially, when somebody dies, their body reaches the temperature of their surroundings. And so if somebody's body is not yet at that temperature, you can potentially back calculate and work out, based on ambient temperature and things like that, when they may have been alive. There's rigor mortis, or the stiffening of a body when somebody dies, which people might be familiar with. And so after death, muscle stiffness happens because of depletion of ATP. But it follows a predictable timeline, and it kind of becomes fully developed at about 12 hours post-mortem. And then at 24 to 48 hours, as decomposition starts, rigor mortis goes away again. So it's possible, based on stages of rigor, to be able to have an estimate of when somebody was alive. And then there's livor mortis, or blood pooling. And once the heart stops, blood starts settling in the lowest parts of the body because of the effect of gravity. And it can cause a purplish-red discoloration. And again, this follows a timeline. It starts between half an hour and 2 hours after death, and then kind of develops over a period of time. And this is useful if you've ever seen the crime dramas where they realise that somebody may have died elsewhere, because once it's fixed, it doesn't go away. So you can kind of see that somebody may have died in a different position to how they were found.
Other ways that you can estimate post-mortem intervals, and you might have seen in a couple of the crime dramas, is forensic entomology, or looking at the life cycle of insects on a body, which can be used to help estimate like how long someone's been there and when they died. Also, the measurement of potassium in vitreous humour, which is a fluid found in your eye that is known to increase after death and could also be used as a marker for post-mortem interval.
These estimates are all incredibly crude. There are so many factors that need to be taken into account to try and estimate the post-mortem interval. And as a result, the actual error on these calculations is exceptionally high. So, Rebecca, why do we as toxicologists care about post-mortem interval?
As toxicologists, when we're looking at different drug concentrations, there are many factors that can influence the concentration of drugs we're looking at. And with a post-mortem interval, you will get like redistribution of certain drugs depending on a variety of factors, and that can affect the concentration that we're measuring. And this may not necessarily be reflective of what was present in the blood at the time of someone's death.
So when you say redistribution, let's clarify what we mean. So the movement of drugs within a body from a high concentration to a lower concentration. So typically this would be parts of the body like the liver, for example, where a particular drug might end up getting sequestered during life, and then in death, that drug then moves into typically the blood, where we then go and measure it.
Yeah, so as Rob's mentioned, you will get passive diffusion post-mortem of certain drugs. As a result of a lack of oxygen, you won't be producing any ATP. You then can't maintain your concentration gradients that were present during life. So drugs will move from an area of high concentration to an area of low concentration. Also, as a result of decomposition processes, you lose your membrane integrity. You'll also get release of intracellular enzymes and components associated with anaerobic respiration because you've not got oxygen moving throughout the body. So things like lactate and formic acid, which can lower your blood pH and make it more acidic, which can also complicate things when interpreting drugs.
And not all drugs behave in the same way, do they? So this is very much drug specific as well, which makes things even harder.
So you can also get accumulation of drugs in certain areas of the body, and these are termed drug reservoirs. And during the post-mortem interval, because this is an area of very highly concentrated drugs, those drugs will then diffuse into local blood vessels, for example. So some drugs, like tricyclic antidepressants, can accumulate in the liver, and therefore you may end up with a higher concentration in the blood vessels surrounding the liver versus other peripheral sites. Same with things like digoxin, can be found quite highly concentrated in the heart. So again, like cardiac blood will have a higher concentration than peripheral surroundings. And things like ethanol, if someone has been drinking before they've died, the stomach will obviously be full of ethanol, and that will again diffuse out of the stomach into local vessels, which is why we generally like using very peripheral sites like femoral blood when we're interpreting.
And some drugs behave differently from others, don't they?
Yeah.
Drugs which are very lipophilic or fat-soluble tend to be more prone to redistribution than more polar or water-soluble drugs. So a couple of examples: antipsychotic clozapine, very, very prone to post-mortem redistribution. Drugs like morphine, much more polar, much less prone to post-mortem redistribution.
And interestingly, drugs can continue to be metabolized after death as well, which can complicate things further. So one example is amitriptyline can be converted to nortriptyline in the lung, where there's a very high concentration of the enzymes required for this. And as well, some drugs have really short half-lives. So by the time we get a sample, drugs may not be present. So for example, when looking at heroin, 6-am has a very, very short half-life of around 30 minutes. So we don't often see it in post-mortem samples, and that can complicate things when trying to identify whether someone's taken heroin or not. So bacteria that are present in the body can also complicate things further. So you can get post-mortem production of ethanol through the fermentation of glucose. So it might look like someone's had alcohol before they've died, when in reality they might not have, and it might just be post-mortem production. That's something that we need to consider as toxicologists when we're writing up our reports. And you can also get bioconversion of nitrobenzodiazepines like flunitrazepam, clonazepam, by a variety of bacteria, and this will change depending on pH and temperature.
So in other words, there are a lot of factors that can affect drug concentrations during that post-mortem interval. A lot of these are going to be dependent on when that person died relative to collection of the sample. Therefore, if we've got a good way of estimating post-mortem interval, as toxicologists, that could be really helpful, right?
Yeah. So many other people have identified the fact that having a more accurate estimation of when someone has died and that post-mortem interval might be really useful for a variety of different reasons that we've already touched on. So we're going to look at a meta-analysis paper that looked at mass spectrometry-based approaches for estimating the post-mortem interval using proteomics, metabolomics, and lipidomics. So that's basically looking at the different proteins, metabolites, and lipids that are available and seeing whether these markers change over time in the post-mortem interval.
And omics approaches were quite popular 10, 15 years ago, weren't they? They're untargeted approaches using large data sets.
Yep.
You don't know what you're looking for. You're just looking for patterns between two different groups and analysing it and seeing if you can find a marker.
So this systematic review included 26 studies that met their inclusion criteria, and each of the papers had a variety of different approaches. So 17 of them were looking at proteomic approaches, 9 were looking at metabolomics, 2 were looking at lipidomics, and 1 was kind of looking at all 3 sort of methods. They all focused on different periods of time in terms of post-mortem interval, so both short, intermediate and long. 11 of these were studying proteomics, metabolomics and lipidomics in human tissues, 18 of these in animal models. So there is that kind of human versus animal difference that needs to be considered when looking at this sort of data. And they were looking in a variety of tissues as well. So for their short-term post-mortem interval estimations, they were looking at things like skeletal muscle, cardiac blood, lungs, kidneys, liver. For medium post-mortem intervals, they're also looking at spleen, tissues from the upper arm, the lower abdomen, and then for long post-mortem intervals, they were mainly focusing on skeletal muscle and bone, which unfortunately, when you're looking at a very long post-mortem interval, that might be all that remains. And 11 of these papers produced a predictive model for post-mortem intervals based on the markers that they were looking at.
So what did these studies find?
So I'm going to split this into the short, intermediate and long post-mortem intervals. So a short post-mortem interval was defined as a post-mortem interval less than 7 days. The proteomics approaches from these different papers kind of focused on muscle tissue analysis, and they found a variety of proteins that decreased predictably in the first 24 hours. So EEF1A2 was one that they picked out, and this decreased predictably in the first 96 hours after death. Some proteins can become upregulated, so increase as a result of hypoxia and processes surrounding that, so like inflammation, oxidative cellular responses, apoptosis. Most of the metabolomics data for the short postmortem interval kind of focused on rats. They were looking at cardiac blood and found 9 metabolites that could be used as a predictive marker in the first 24 hours. So xanthine and isoleucine were markers that they'd picked out. In skeletal muscle, they found 59 compounds with a strong correlation of estimating postmortem interval in up to a week. Yes, there's lots of different compounds that could be used in this sort of short time, but none are kind of in routine use at the moment. Moving on to the intermediate postmortem interval, which they class between 7 and 120 days.
That's quite a difference, isn't it?
Yes.
Quite a wide range.
And for these studies, they seem to focus on animal decomposition fluids, and there were consistent alterations in a variety of peptides derived from things like hemoglobin subunits, creatine kinase, lactate dehydrogenase. A lot of the things they found with these markers is that they're sex and BMI specific. So that kind of complicates interpretation quite a lot. So targeted reference ranges would need to kind of be established for a very wide range of individuals to kind of make this sort of approach useful.
And collecting that data is going to be a nightmare, right?
So complicated. And this was the only post-mortem interval time point they looked at sort of lipidomics. So stearic acid and palmitic acid increase during specific decomposition stages. And interestingly, unsaturated fatty acids can mark the transition between different decomposition stages. So it might be quite useful as a general marker for post-mortem intervals. In the long post-mortem interval, so that was anything greater than 120 days, they're mostly looking at bone tissue, as we said before, this might be all that's left to analyse. And there are a variety of proteins, like histone proteins, that can help discriminate between fresh and skeletonized bone, which can also be quite a useful marker. And there are also a variety of collagen-based proteins that they looked at that also seem to work. But as far as I could find from my reading, there isn't an established marker that is being used to more accurately estimate the postmortem interval, because there are just so many different factors that can complicate it. So you've got sex, age, BMI as factors. You've got people's medical histories and comorbidities that are going on, people's individual genetic makeup. So you might have people who produce more of one protein or less of one, which complicates things further. And then you've got things like environmental conditions, so where the body's been, that will also influence how these proteins and metabolites and things change.
So these are all sort of omics studies, and they've all gone and identified a few possible markers. Did they have any opinion on whether any of these show promise?
So basically from this review, they concluded that this could potentially be a great marker of post-mortem interval if we find the right marker. But from this study, there are so many different factors that kind of prohibit a conclusion of one over the other being drawn. Different sample sizes, animals versus human models, things like that. And then there are also all the environmental and like individual aspects that I've already mentioned that can affect these markers as well. So it's not quite as clear cut as if this marker goes up or down, then that's how long someone's been dead for. There are many other factors that would have to be built into an algorithm to kind of account for different changes. Whether that's even a thing that's possible to do, I'm not too sure.
I'm just thinking about sort of large AI models and things like that, whether there is possibly a multi-factor model that could be derived that would actually take into account these factors and tell you when it's likely somebody died. I mean, as toxicologists, it would be great, wouldn't it, to have a nice little analyte we could measure, and then we could go, okay, it was this many days, therefore we need to adjust all of this drug by this amount. I mean, that would be the dream, right?
Oh, it'd be lovely.
But in reality, it's just really messy. You've just got these huge uncertainties. And so, at least currently, it just feels like a fairy tale.
It does, yeah. Maybe one day we'll have something that can help us more accurately determine. And as you said, AI might be the answer with something like this. There's so much data that would need to be processed that the use of AI might be the key to kind of unlocking this.
But even then, AI is only as good as the information you can feed it.
Yeah.
And so if you're relying on existing, it wouldn't be ethical to kill humans and study the post-mortem interval, would it?
No, it
wouldn't. So you already effectively, even if you're going to use AI, training AI with knowledge that's uncertain anyway.
Yeah, that's very true.
So in summary, post-mortem interval estimation is sort of a dark art, borderline witchcraft. Certainly not like it is on the drama shows where they look at somebody and go, they've been dead for 34.6 hours. And as toxicologists, it would be really, really helpful to have a nice marker or three that we could measure to tell us what that interval would be. But as it stands currently, it doesn't look like one exists, at least for now. Possibly with AI, that might change. So I hope you've enjoyed listening to us this week. Do give us a like and a follow on Spotify or Instagram or any of the other. Rate us on Apple Podcasts. That would be great.
You can find us on Instagram at @the_tox_lab. We'd love to hear from you, so feel free to comment on any of our posts about any of our episodes. You can send us a message. We also now have an email. So if you don't do social media and want to get in touch with recommendations for future episodes, you can email us at thetoxlab@gmail.com. We'd love to hear from you. Any feedback, any content you'd like us to cover in the future would all be greatly appreciated. Thank you so much for listening. Hope you all had a wonderful week, and we'll see you next time. Bye.